All right. I hope you can see my screen. So this session, I'm going to talk and introduce you to tools for protein exploration. These are some of the tools that we have implemented in GlyGen. And my name is Jeet Vora. I'm a data manager, scientific coordinator for GlyGen. And I'm primarily involved in integrating protein data and also implement the protein tools that are useful to the community in GlyGen. So today's in my session, I'm going to cover a BLAST, which is a very popular tool. We have implemented a version at our end. I'm going to also talk about GlyGen Mapper, which is essentially the ID mapper. And I'm going to talk about GlyGen Site Search, which is an interesting and unique feature to GlyGen. And after that, we are going to do two exercises based on BLAST and GlyGen Site Search. So with this, I'm going to now talk about BLAST. I'm sure that many of us here today have, during their academic course or in their academics, they have used BLAST for homology, for detection of similar proteins, for taxonomy classification and whatnot. So let me give you a little bit background about BLAST. So BLAST was developed in the early 1990s, and the lead developer was a researcher, Stephen Alchul, here at NIH. He was with the National Center for Biotechnology Information, which we all know as NCBI. So that was when this was developed, and the reason for it was to address the need for efficient and accurate sequence comparison methods, because at that time, the genomics and bioinformatics was burgeoning and blooming, and people wanted to have such tool where they could make sequence comparison. And by the way, the BLAST stands for Basic Local Alignment Search Tool. And this is, as I mentioned earlier, one of the most popular tool in bioinformatics, and it's widely used. I think some years ago, I heard from someone that like currently the Nobel Prizes are not given to people in the field of bioinformatics per se, but if it were to be given to anyone, I think the BLAST team, stands on the top because that has transformed the field to a great extent. Now, as I mentioned, it is used for searching sequence databases for sequence that are similar to your input sequence, or as you know, a query sequence. So you put in a query sequence and it will search its sequence database. And these are nucleotide protein sequence in FASTA format, and you'll get some matching sequence based on the query sequence. So BLAST, it can be used to compare different biological sequences, such as nucleotide sequences, DNA or RNA, or protein sequences. And, it can be, the BLAST can work against the standard database, or you can also have your own database and find homologous sequences which are similar in their nature and sequence. So how does the BLAST algorithm work? It works by finding regions of local similarity between sequences. So it's the BLAST breaks down the input sequence into multiple smaller chunks, which is called as words, or shorter of fragments or peptides, and then it matches for a corresponding similar word or the sequence in the databases. And then once it finds it, it provides you some information like score, and identities, gaps, and all of that, which I'm going to talk later. And from that, you can see, what sequence or what hit is similar to the query sequence found in the database. Now, BLAST has a few different flavors, and they are used for different purposes. The most popular are BLAST-N and BLAST-P. The BLAST-N is used to compare nucleotide sequences against the nucleotide sequence database. So if you have a nucleotide sequence, you can BLAST using the BLAST-N. For protein, you use BLAST-P, so that compares your protein sequences to the protein sequence database. And the other two are closely related. So the BLAST-X, it's used to compare your nucleotide query sequence against a protein sequence database, and the T-BLAST-N or T-BLAST-N is used to query your protein sequence against a nucleotide sequence database. And the other one is T-BLAST-X that compares a nucleotide sequence to reading frames that's translated in all reading frames against a nucleotide sequence database. So these are the few flavors of BLAST. And in GlyGen, we have implemented the BLAST-P. So how you can access BLAST using their web interface. They have a well-developed interface with tons of features, and it's intuitive. It takes like I would say a few tries to get everything in place and order, and you can run your BLAST. Also, if you are programmatically savvy, you can also run it via APIs, and they also have a BLAST implementation in cloud, which you can make use of in your research or analysis. So just want to summarize. So BLAST is a very powerful and widely used tool in informatics, and it's used primarily for sequence similarity searching, homology detection, functional annotation of biological sequences, and it has played an important role and provided research applications in genetics, genomics, molecular biology, and related fields, including bioinformatics. So this is what the BLAST looks like, implemented by NCBI under National Library of Medicine. And as I mentioned, they have nucleotide BLAST, the BLAST-X, T-BLAST-N, and protein BLAST. I'm not going to go into detail, but this is just to give you an idea of what the interface is like. And then there are some other features and tools with them. I mentioned you can download your BLAST, you can run it locally. You have a BLAST API, you can run it BLAST in the cloud. And there are other features and tools also recently added to this BLAST web page maintained by NCBI. So now let's talk about GlyGen BLAST, and I'm going to show you the functioning of GlyGen BLAST is similar. There are a few nuances, few things that are different from the BLAST implementation at NCBI. So we're going to look at that. So how do you access BLAST from GlyGen? So there are a couple of ways to do that. You go to the GlyGen homepage, just glygen.org, and you click on Tools. So this is the Tools, and from there, I'm just going to select laser pointer. So from Tools, you can select the GlyGen BLAST, and you can open that. Or there is another way that you can do is you can select on Explore or click on Explore from one of these cards which are located on the home page. And once you do that, the BLAST tool interface will launch. Now, if you see, this is very simple interface, very few options to put in, and it's very easy to follow and work with it. So how it works is either you provide it with a UniProt KB accession, or you provide it with a sequence. Now, we can, if you want to explore, you can just use the example accession, which is the protein accession for HGF, hepatocyte growth factor. And once you click on it, the sequence, and when you click on retrieve sequence, the sequence automatically populates in the sequence text. And then if you don't want to play around with or see options what are there in the target database. You want to keep the evalue as is, number of hits, proteins as is. You can just click on submit. And I'm going to talk a little bit about evalue and other nuances of BLAST. And when you do that, it runs. As I mentioned, if you don't know the UniProt-KB accession, you just have your peptide sequence or protein sequence in a FASTA format. You can add it, and you can click on submit for BLAST to work. So talking about the target DB, so as I mentioned, we have implemented a version of BLAST in GlyGen, and how it differs from the original BLAST is our sequence database is limited, meaning that we don't have all the organisms in GlyGen, but we have select few well-annotated organisms in GlyGen. And you can run a BLAST, run your query sequence against the target DB containing these few organisms. So Dictyostelium, Drosophila, we have some viruses, of course, Homo sapiens, Musculus, Rattus, Norvegicus. We have yeast, so on and so forth. So you can either use the default option as all species, or you can select a particular target database of sequence. When you do that, I'm going to keep the e-value as is. It's the preferred e-value when doing BLAST. So I'm going to keep it as is. The max number of hits is it's the proteins that it needs to search against, or to have a limit of proteins it should show as a result. That's 250. I'm going to keep it as is. And then I'm going to click on submit. And what happens is the BLAST is working. So you get a message waiting for the job to complete. It's querying your sequence against the database. And once it is done, it will provide you with a list page or a result page, I would say, with the summary of what you've done. So the job type is BLAST P, BLAST protein. This was the sequence that was entered, the target DB, the e-value, and the max number of hits of proteins. And if you change your mind, if you want to do, you can always click on modify request and start again. So when you click on that, you would see a table kind of appears along with the summary. And the first tab that you see is the result. So on the result, you will see some high scoring pairs. And these are, the pairs against or the pairs that were found when the BLAST did a match with the query. When the BLAST ran your query sequence against its databases. So this table is also simple. You have the UniProt-KB accession. You have the position where it matches, the UniProt-KB ID, the protein name, the gene name, standard things that you work with. Of course, the organism to give you a snapshot of the organism information that BLAST has found, and the e-value that's present in this column, and the identities. And also going to talk about the identities. But right now, 728 out of 728, which is the length of the HGF protein. And this was the hit, of course, because ours was the query sequence. And this is what it found. But these are the other other information or other proteins and the other sequences that were similar to our query sequence. And you can also use the sort option to sort the information that you see on your screen. So I'm going to click on one of the alignment. So to do that, you can either click on this two-way arrow, and it will give you a page where it shows the alignment. Now, I wanted to make one thing very clear. If you run a BLAST, the original BLAST, it will also map to the isoform. But for GlyGen, we are only mapping it to the canonicals. If you don't know what a canonical is a sequence. It's a concept by UniProt-KB, where most of the annotations are available, or the sequence where they have the annotations available, and that's what they share. So every entry or UniProt ID of a UniProt will have a designated canonical. It can be both Swissprot or Tramble. So that's, I wanted to clarify. We are also in the process of adding isoforms. So you can also see the isoforms of a given UniProt KB entry. Okay, so when I click on the two-way arrow, you come to this page where you see the alignment. And the alignment, the page offers you, the select protein ID. Since I selected plasminogen for mouse, it shows here, but you can also change that later. And it provides some information about the protein again, and with some additional information. So it shows you your UniProt KB accession, protein name, gene name, the organism, and some of the values about some of the results from BLAST. And these are the e-values, score, positives, identities, gaps, and the method that it used, which is the compositional matrix adjust. And here in this grade portion or the alignment portion, you will see your query sequence. This is your query sequence. And this is where they have mapped. I'm just showing the partial view here. And this is your query mapping to the plasminogen of mouse. And these are the conserved sites. If you know alignment, you would know. Plus is these amino acids are similar in their properties. And when there is nothing, there is no match or similar amino acids found, it is usually blank. And when there is a dash, it means there is an insertion or a deletion that has occurred. So this is how you can look at the alignment based on your query input query sequence and the selected high scoring pair in the result. You can also directly click on alignments and come to this page. And as I mentioned, you can click on the select protein ID to check other options and see if you are interested in knowing the alignment with few of these options that it has matched. One of the interesting feature that we have implemented is the site feature, where the annotations, when you click on the annotations, it gets highlighted. Now, what it shows is there are 4 n-linked sites. And so these are the red ones, the red underline. These are the n-link sites. And also it's an n-link site, and therefore it has the n-sequon. So when you click on the sequon, it also gets highlighted. Then you have O-link sites, which is by a blue underline. So you see here the threonine is underlined. Then other than N-linked, O-linked, we also have SNV. So there are 13 such sites that are known to have a single nucleotide polymorphism. Then we also have phosphorylation and glycation. Since no annotation is available for these two annotation or PTMs, we do not show it over here. The other interesting feature you can do is you can also do a custom search. So you can put in a pattern, or you can put in a short peptide, and it will find this the input peptide or input motif or input sequence in the sequence. One thing I would like to point out, so it is possible that based the annotations are it will be shown, of course, for the proteins that are in GlyGen's database. So only those will get highlighted. There is currently no option to add additional highlight to the query sequence. But if you do like custom, it will highlight few of those positions where it found the sequon or the peptide in the query sequence. So again, an interesting feature to take advantage of. And then we also have in the text view, if you prefer to look it in a text view, we have also implemented that, and this can also be like downloaded. You have the isoform, which is the canonical isoform, because canonical is part of the isoform of the protein entry. So uniprotic session, the protein name, or the protein header for the sequence. Then you have the bit score and the e-value. You can download and play with it, filter it, to get the best match, or to select the sequence of your interest. Okay, so I talked about e-value, bit scores, positive identities, and gaps. So what in essentiality it means? So I'm going to go over those and try to provide some context. So e-value is it's a statistical number, or it's the value which indicates how reliable your match is, or how meaningful, or how biological meaning your match is. So it tells you, as I mentioned, how significant or reliable your match is. And as I mentioned, the lower the e-value indicates a higher significance, or if you put lower e-value, meaning that you won't get any hits that are occurring just by a random chance. So your result, or the result that it will show you will be very significant and meaningful. So that's about e-value. Bit score, it refers to the statistical measure of the significance of the sequence alignments. It's a score that will again provide you with information of how significant your alignment is, or the result that it is found, if you want to use that. And usually the higher the bit score is, the better your alignment is. Then I want to talk about the positives and identities. These are similar, but um, uh, these are like, but they do have some nuances, and you can differentiate based on the nuances. So positive refers to identical or conserved positions in the alignment. And positives, they do not necessarily indicate these are exact matches. So, and asparagine and asparagine, if it's found in your query sequence as well as the mapped sequence, then they are conserved, or it could have a similar amino acid having similar properties. But there can be also chances where you have where you do not have a conserved one. On the other hand, identities means the any like amino acid nucleotides are identical. The query sequence and the subject sequence, they are completely identical in your result. So that's what the identity is. And gaps refer to the insertion or deletion of nucleotides or amino acids between the query and the subject sequence. So these are the nuances where you have to deal with when looking at the BLAST results. So I'm going to go back again and show you. So the e-value, we have chosen a very low e-value. So our result is significant. Also, the bit score here is quite high compared to the others. The positives, in this case, since I've only chosen the first match, and of course it will match. So the positives, all the amino acids are similar or conserved, and identities say they are the exact match, 100% exact match. And there were 0 gaps, so no insertions or deletions in this particular one. But I showed one more example about plasminogen. In here, the e-value is higher, so it might not be a best hit or best match or best result to consider. But nonetheless, we have got this, and we can look into it more in a detailed fashion. So its score is also relatively lower. The positives are 346 out of 632. Identities is 255, meaning 255 are the exact match. And there is like gap of 8%, 50 amino acids out of 632. So these are a few of the details about BLAST result. So to summarize the BLAST tool, it's used for searching sequence database for sequence that are similar to the given input sequence. It can be used for homology detection, functional annotation, similarity searching, some cases protein detection. That's the main use of it. In GlyGen, we have implemented BLAST P, just that flavor of BLAST. And we offer alignment for canonical proteins for select organism or protein database as few organisms as mentioned. And we have the additional annotation selection feature, which the original doesn't have. So it kind of gives you an idea of what PTMs are there on the subject sequence. So that's the summary. And then we're going to do an exercise at the end for BLAST. If you have not used and if you want to try GlyGen, then that exercise will gives you a hands-on experience. Now, talking to the or coming to the second tool, which is the ID mapping. So what does ID mapping mean? So in biological databases, it means it's process of either linking or converting IDs or accessions from one biological entity. It could be a protein, glycan, a lipid, or from one database to another database. Let's say from UniProt to RefSeq or from UniProt to GeneID, so on and so forth. And why this is important? ID mapping is important in bioinformatics context because it's important to integrate data from different resources, and it kind of facilitates interoperability and cross-referencing. So it's easier to integrate data from different resources if they provide a mapping. And it's a standard mapping, and the accessions IDs relatively don't change that often. So these are a few of the resources that have ID mapping feature. So these are just a few that I am showing you on the screen. So UniProt has one, protein information resource, PIR, then database for annotation, visualization, and integrated discovery. David, they have a version of ID mapping, bacterial and viral bioinformatics resource centers. So they have ID mapping, and then we have also implemented the GlyGen ID mapping. So these are some of the few resources. Each of the resources will have different flavors based on their data and based on their data model. But in a sense, they work in a similar fashion, or the technology behind it, or the concept behind it is similar. So this is what the ID mapping looks like on UniProt. So you can put in your ID mapping here. In the text box, you can also select from database to database, and they offer a varied list of databases, usually the protein databases, in the drop-down option. Then PIR also has a similar interface where you have from ID to ID, and you can put in your IDs. You can also upload a file, and you can select a taxonomy ID, and it will provide the mapping to you. If you are working in bacterial and viral space, then this could be of interest to you. David is also another tool for ID mapping that is, I think, primarily focused on gene. Okay, coming to the GlyGen mapper, how do you access GlyGen mapper? So again, like BLAST, it is located in tools. From the home page, you click on tools and go to select GlyGen mapper, and it will open the GlyGen mapper interface. Or you can also select a card, which is there on the home page, and you can access the ID mapper tool. So this is what the ID mapper or a GlyGen mapper tool looks like. Again, a simple, clean interface with just few options to fill in. The first option is By default, it shows Glycan. So you can click on that, which is the molecule field. We can click on that and it will show you two options. Right now we have Glycan and protein. And I want to mention, like, I think GlyGen is currently the only resource providing ID mapping for a Glycan molecule along with the protein molecule. Then you have a similar option as we saw in Uniprot and in PIR. So you select your from ID, the database for from ID, and then you select. So in GlyGen, yes, you see a bunch of resources that have information related to Glycan, or they have their own IDs for glycans, denoting glycans. And you click on one. So in this case, I'm selecting Glycan. It's the primary repository, or the repository or database that provides accession to Glycan. Yeah, so you click on that, and then you can select the two database from the list. It kind of shows you the same one, and you can select one to get the mapping. So in this case, I'm going to select PubChem compound. And once you fill that, then it will ask you to enter IDs. And keep in mind, all these three or four fields you need to enter or type in for this ID mapper tool to work. So you can click on the examples to try out, or you can enter the IDs that you have. And once you do that, you can click on submit. Alternatively, you can also upload a text file, which can have your accession in. So there has to be like one accession per line and one ID per line, and you can upload that, and it will also take in and perform the ID mapping for you if the ID or accession list is long and you don't want to manually add it in the enter IDs text box. So you click on submit, and you see a list, a result page. So a lot like we saw for BLAST, you'll get a summary of what you performed. So it will also give you some statistics about mapped IDs and unmapped IDs. So in this case, all our 5 Glycan IDs for glycans have been mapped, and there are like 0 IDs, 0 input IDs that were not mapped, or there was any problem with them. So that was the mapping stats. You can also download your mapping. Of course, you would want to work with it. So there are download options. And if there is any other message or unmapped IDs, you can download both the tables, the one that has your mappings and the other one that didn't map. Or you can download an individual table, based on your preference on how you want to work with it. So yeah, that was it. So this is your from ID, the selected database, which is Glycan, and you see these are your entered Glycan accessions. How it performs mapping is it identifies it based on the existing database. When it finds, yes, that is a valid Glycan accession, it is there in GlyGen, it will provide an mapping to PubChem compound, provided the mapping is existing in GlyGen. So keep that in mind. Now, you can also, so one flavor I want to show is, yes, you have entered few of your Glycan information, and you see here I have just put in some gibberish text, ABC 1 to 3, and this is a protein accession. So in case you enter incorrect or invalid accessions, then this is what you would see. So out of those, the 5 that we added, yeah, they were mapped, but there were certain like 1, 2, 3 ABC. It will show you it's an invalid ID. For few of those, it will show you it's a valid ID. This accession is valid, but there is no mapping available from Glycan to PubChem from us. It doesn't exist, so it's saying no mapping found. And again, because this is Glycan, it won't find um, the mapping for protein. So you can't map between a glycan, like most of the resources from glycan, to a protein resource. So that thing needs to be kept in mind. And with this, we have shown you the GlyGen mapper, and this is the summary. It provides glycan and protein ID mapping. We have 42 resources that can be cross-linked or ID mapped for glycan, and 39 resources for protein. And from the protein, some are very organism specific. It could be HGNC for human, MGI mouse, RGD for rat, so on and so forth. You can also enter accessions in the text box or upload a text file with one ID per line. The list page, it provides you at a glance maps, stats about the mapping, and it also provides you with the reason for unmapped IDs. And the result, you can download it and work with it, as you wish. So that was GlyGen Mapper summary. Now the last tool, an interesting tool called as GlyGen Site Search. I'm going to talk about it, and you can use that, especially if you're in the field of proteomics, glycoproteomics, glycomics. This can be a good tool to be used for further exploration. So what is a GlyGen site search? So in GlyGen, we refer it to the search functionality available on GlyGen, of course, that allows users to query the database for very specific information related to site or amino acid of interest. That has to be on a protein sequence, and that site has to be associated with one of the annotations, or we can also select several annotations that are available in GlyGen, and the list of GlyGen annotation is glycation, glycosylation, mutagenesis, phosphorylation, and SNV. So based on this annotation, you can do your site search. How do you go to the site search? So on the GlyGen homepage, you have on the menu, you have Explore option. When you click on that, you will see a bunch of options, glycan search, protein search, and the third one you'll find is for the site search. So you click on that, and the site search interface opens up or displays on your screen. The other way is, of course, you can do that. You can access site search from the card on the homepage. When you click on Explore, the site search also opens, and this is the interface that you see. Of course, we have some tutorials on how to use that. You can also look at those tutorials and see what you can do. So we have like few options here, and they're very easy. You just need to spend a couple of minutes just to understand. And we have some help. You see this question mark, so there's some help text that will allow you to understand what that field means and how you can enter the information. So the first thing, by the way, I just want to mention, none of these are required. You can just click on search protein site, and it will give you a list of all the protein sites that have the annotations, the 5 annotations across the GlyGen database. Okay, so you can, if you want to search for specific amino acid in the proteins in GlyGen, you can click on that. Once you click, it will show you the list of amino acids, and you can select the amino acid. So in this case, I'm selecting serine, and then you can also, as I mentioned, you can directly do search, and it will show you proteins that have serine as amino acid, but that's relatively an easy search. But if you want to do a little bit more advanced, then you can also select annotation type. In this case, the 5 annotations are displayed, and you can select based on your preference. So I'm selected glycosylation and phosphorylation. So it's going to now search for all proteins that have serine, but on that serine, there is an annotation or that serine is glycosylated or phosphorylated. that's what it's going to search for. You can check the option as an OR operator. You can select any of that. So I'm keeping it as default. And then so based on the operators, you'll get different results. And then this is the next field. What you can enter is the neighbors. So you have this serine on a protein which is glycosylated and phosphorylated. Is there anything else you want to see that is in the vicinity of that serine? So, when you click on that, you get an option. Well, okay. Um, yeah, you get an option about what you want. So we're going to try this, neighbors and pattern in the exercise, but I just want to mention when you click on that, it will show you 5 annotations. So let's say you want to see or you want to know whether there is any mutation site or any mutagenesis experiment that has conducted, let's say from 5 amino acids, it can be equal to or less than 5 amino acids, or greater than 5 amino acids around the serine. So you can specify the distance, and you can also specify the operator. And to make it more complicated, or I would say interesting, you can also add one more search, which is the pattern. So this pattern is where you can enter a peptide or a peptide sequence, a short sequence, or a motif, or a sequence. You can specify a position, and you can specify where where this tool needs to find that peptide in the protein that it will be find. So it could be either C terminal, which is downstream, or it could be upstream, which is the N terminal. And you can specify a position. So I want to check whether there is an n-link sequence. Let's assume n x t. I want to see if any protein has this n sequence, which is downstream, at the C terminal, and 5 or let's say 10 amino acids away, on that protein sequence. So when you enter that, it will also take into that consideration and try to match it and provide you with a result. And as I mentioned, you are not required to fill in all the fields. You can just go to search protein site, and it will give you a list of all the sites that are present in GlyGen, and you can also do some filtration and all of that, which I'm going to talk about. So I selected, in the summary, I selected the annotation type as glycosylation and phosphorylation, and I selected amino acid as serine. And this is what the result list page I would see when the search has been executed. So you'll see there are like 2,737 results where a serine serine is found, the protein entries, and these serines are glycosylated and phosphorylated. Also, since there can be more than one serine for a given protein, so there could be the same protein but with a different start position and position, basically a site appearing in the result. So you need to take that also into consideration. Then, since I wanted glycosylation phosphorylation, so it will tell you, yes, these results have both glycosylation and phosphorylation, and some other information also you can see along with glycosylation and phosphorylation. You can also try to filter, based on glycosylation type, amino acid, SNV, organism, so on and so forth. And if you want to work with this table, you can download it in your local computer and work with it. So that's what it shows. Now, you see there are two options highlighted here. One is the UniProt KB accession and the start position and end position. So if you click on that, it will take you to the site-specific detail page. So I clicked on serine 74, and this particular page has all the information related to that site only. But in the beginning part, it will show me some general information about the protein that has that site S serine 74. So it's a keratin type 2 cytoskeletal 8 protein. And the serine 74, it has glycosylation, mutagenesis, SNB, and phosphorylation information. So it's O-linked site, serine 74. It has also phosphorylation information on 74. I'm not showing the other part, but you'll also see the SNV or any mutagenesis information on that page. So this was our simple search. Now to summarize, the GlyGen site search, it provides, as I mentioned, very unique site searching capability. I have not seen it in any other resources that I have explored. So it's quite interesting and unique. Right now, we have 5 annotations or PTMs per se, which is glycosylation, phosphorylation, SNV, mutagenesis, glycation. Actually, 3 PTMs and the other 2 are protein modifications. And then I also want to point out that you can also search for neighboring annotations that are around the vicinity of your site having a particular annotation. And it also allows you to search for a short peptide at a specific distance from the site of a selected annotation. So give that a try if you have a research use case. And we will also be doing an exercise on this in the next few minutes. And you will be able to explore the tool yourself. So I have already in the chat added a hands-on exercise where I have the question, and you can do that. So there are 2 hands-on exercise. One is for BLAST and one is for the site search. So this is the link. I'm going to put that link again in the chat. So if you are okay, so everyone is meeting. I've added that link. So there is a hands-on exercise where you can explore this. So now we have almost around 10 minutes or so. So 5 minutes each. We can explore one hands-on exercise and do that. You can also do it later. I'm going to also quickly show some of the things here on the live interface, and we can do that. And also after that, if you have any questions, I'm happy to answer any questions, queries, or concerns. So I hope you can access the hands-on material. I'm also going to share my screen with the first exercise. So this is about GlyGen BLAST. So a task is a use case based task. So it's from a serum sample of a liver cancer patient, a certain protein was found at a much higher level than in normal adults. Using biochemical techniques, the protein could be separated, sequenced, and identified. And this protein was alpha-fetoprotein. Researchers discovered that the alpha-fetoprotein can represent as a most prominent oncobiomarker widely used in diagnosis of the cancer. Now, these researchers are interested in finding any other human proteins that are similar to alpha-fetoprotein in terms of functions and roles, and if they can find a new biomarker, how would they find similar proteins? So, of course, you would have to use GlyGen BLAST, but before you use that, you need to have a protein fastest sequence for alpha-fetoprotein or a UniProt KB accession. So I'm going to give you a few minutes for you to try it out yourself. There is also solution that is shared with you. You can follow that and do your blast search. This is just a use case example. Your research might have a different use case, and you can try to use GlyGen and see what similar proteins you find. And yes, do let us know if you have any questions about it. One thing, because we have a few people here, and it's the GlyGen server, it takes in the request, the API request. So it can take like from few seconds to few minutes to give the result. So bear with it. All right, I am going to.
Jeet, you have a question in the Q&A, which I'm guessing might be well answered by the results of the blast search.
Okay. Is there a way, this is from Caitlin, is there a way to get the SNPs associated with variation from mutation? So these, so we have single nucleotide variations in GlyGen. These are point mutation. And yes, there is a way you can get those. Now, there are a couple of ways. If you want the entire data set, and this is specifically for human at the moment, you can get them through data set, or if the other way is to use the site search and put in SNV, and you'll get all the sites that have these SNV mutations. Can show that probably. So I'm going to click on SNV if that's what you're interested. You click on search protein site.
Hey, is there a way to get it for a specific gene?
For a specific gene, I mean, not from the site search page, but the other option is to go to a detail page and check that. So, for example, I would do this. So this is like you've got more than 100K results because from the Homo sapiens you have 100K entries with that SNV information. But for a specific one, either you can use the protein search. Go to the details page for the protein. And from there, you can go to the the SNV section, so single nucleotide variation. So you'll get like two tabs. One are the mutations that are associated with the disease, and the other one is the non-associated. In the disease, you can have a somatic or a germline mutation, and it will show you some annotations to also tell you whether it's an O glycoside loss or N glycoside loss, and it will provide you some of the information. So if you have a specific gene or so, I mean, this you will have to do one by one. But if you are tech savvy, you can also use Sparkle endpoint and do a Sparkle query where you can get where you can put in your genes of interest and get the information for the single nucleotide variation available in GlyGen.
This is perfect. Thanks.
Okay. Yeah, sounds good. So we have like 5 more minutes, and I will be showing that last thing. So yeah, we have the accession. We'll go to GlyGen BLAST. I'm going to put in the sequence, and this is -1. It represents the canonical. You can either use that, or it can be just the UniProt sequence. Now it has retrieved the sequence, and we are going to keep all of the example as is. And I think, yeah, we for the target DB, we want to choose Homo sapiens because we're interested in finding similar proteins in humans. I'm going to click on submit. The server is going to work on it, send in the API request, and provide and perform the blast and provide the results. All right. Okay, so it has found 6 high scoring pairs, and in all, I see like 3 proteins that are similar to our alpha fetoprotein. But if you look at a glance, then, I mean, the E values are quite high. So in terms of similarity, they're not that similar. But to an extent, you can say, they have similar based on the identities. It is 40% for albumin and alphamine, whereas the identity percentage drops for the vitamin D binding protein. So if you closely look at these proteins and if you do a research, they are from the same protein family, and thus they're bound to be similar in some aspect. And then, you can click on one of the alignment to see how it looks like. You click on the alignment and you get other score. Of course, this is not very highly significant, but nonetheless, you have some values that will allow you to make decision. And you can play around with N-link tooling sites to see, if there are any conserved sites or conserved peptides in your sequence or subject sequence. So yeah, none of the sequence doesn't have an N site. So I mean, this is one of the way you can explore further. You can click on this protein and you can find great detail about this particular protein, which is albumin, a non-protein. So GlyGen will have information about function, nucleotide variation. There is a mutagenesis experiment on it. Then it's any disease association, it will show you any biomarkers. It will show you here. So there are biomarkers for this albumin protein as well, any expression information, so on and so forth. So this is what you can do. Now, if you have just have a minute or so, so the next exercises also are relatively easier to follow. And you can do that. So what it says is, what the task is, find proteins that are N-glycosylated with phosphorylated sites greater than or equal to 5 amino acid in distance of the N-glycosylated sites. And they also have a N-sequon, the NXT is the sequon, at 5 amino acid distance towards the N-terminal. So this is a task. How we're going to do is you will have to go to site search and perform that. So I'm going to move this aside. I will go to my site search. Okay, in this we have selected N-link. It's supposed to be N-link site, so we'll look for asparagine. So NASN asparagine, we found that. That is our first condition. It has the second condition is based on the annotation type. It has to be glycosylated as well as phosphorylated. The annotation has to be and then the neighbors is I think we mentioned it has to be. Let me check that again, just to double check. Okay, so they just have to be. Of course, N-glycosylation and phosphorylation do not occur on the same site. In the neighbor, we want to have phosphorylation, which is 5 amino acid, and which is more or less, greater. And in the pattern, we want it at the N-terminal. Specified position is 5 amino acid, and the sequence that we want to search for is NXT. And we click on search protein site. And once you click on it, it will try to find the site that fulfills all this condition. Okay, so I think I will have to modify it. Oh no, it's perfect. So based on the conditions, it found us like three protein. This is one protein, but based on the organism, it provides three results. So one is for Homo sapiens, mus musculus, and Ratus norvegicus, and the protein is contacting 6, and this is the result. So now we are going to see uh, uh, whether the result has fulfilled all the conditions or not. You can go here by clicking on start and end position, or you can go to UniProt KBX session, and from there, you can go to the sequence option where you can visually see whether it has found the correct match or not. So when you click on the sequence, it will show up here. And what I'm going to do is I'm going to click on N-link sites. It has to be phosphorylated. And there was a sequon NXT. Okay, and I'm going to click on it. So the position that it showed us was 865. So that is an so this is the position for the N-linked site. This site is 865. And if you see, there is a phosphorylation site. It's a phosphotyrosine, which is greater than 5 amino acid in the distance in the vicinity of our N site. And then it found an NXT sequon, which was at a distance of 5 amino acid, and that is what we specified. So if and towards the N terminal, so if you see 1, 2, 3, 4, and 5, so this is where it found that starting sequon. So it provides site search provides an interesting feature. If you just have a sequon, a motif, let's say for C man oscillation, or there are some polling like oscillation, which like fugue oscillation that are around a particular motif or on the motif, we can use this site search and try to find proteins that fulfill this condition.